Hello, I'm Alex Weaver, and I'm here to speak to you about my research on Arachidib-1 as a possible treatment for triple-negative breast cancer. Triple-negative breast cancer accounts for 10 to 50% of all breast cancer cases. It's invasive, spreads quickly, and has worse patient prognoses than normal breast cancer. Arachidib-1, or A1, is a natural plant compound produced by peanut hair root systems. This compound is then extracted and purified via HPLC, as you can see here in Figure 1. The goal of this project and the hypothesis was that Arachidib-1 will inhibit the proliferation of triple-negative breast cancer by inducing apoptosis-related proteins. To test this, we extracted and purified Arachidib-1, examined the cytotoxic effects of Arachidib-1, measured apoptosis activity, and observed the level of apoptosis-related proteins by Western blots. Figure 3 is an example of our experimental design, which I'll now go through step by step. Figure 4 is the first thing we did, which is observe the effects of Arachidib-1 on triple-negative breast cancer proliferation. We used two triple-negative breast cancer cell lines throughout this entire study, MDMB-231 and MDMB-436. These cell lines were treated with various tenfold concentrations of Arachidib-1 for 3 time points, 24, 48, and 72 hours. From this, we were able to find an IC50 value and move on to further studies. The next thing we did was observe the cytotoxicity effects on different cell lines, the two previously mentioned cancer cell lines, as well as a non-cancerous MCF10A. Cells were treated with 1 micromolar Arachidib-1 at the same 3 time points, and we found that there were significant cytotoxic effects on both cancer cell lines, while Arachidib-1 had no significant cytotoxic effects on non-cancerous MCF10A. Next, we moved on to looking for apoptosis induction via staining and flow cytometry. For this, we treated both cancer cell lines with 0.1 and 1 micromolar Arachidib-1 for the same 3 mentioned time points, and we found that Arachidib-1 had apoptotic effects on both cancer cell lines, having a dose-dependent effect on MDMA-231 and a late apoptotic effect on 436. Next, we move on looking at cell cycle arrest. So for this, we used the same cancer cell lines, the same treatments, and the same time points, and found that using PI staining and flow cytometry, there's a significant dose-dependent increase in G2 M-phase cell cycle arrest leading to apoptosis and increasing Arachidib-1 concentration. The next thing we looked at was trying to figure out if it was intrinsic or extrinsic apoptosis that these cells were undergoing. For this, we treated the cells with 0.1 and 1 micromolar Arachidib-1, harvested cells and ran Western blots, and then we were able to make densitometric comparisons. From these densitometric comparisons, we found that cells treated with Arachidib-1 had an increase in activated caspase-9, activated PARP, and a decrease in anti-apoptosis protein survivin that led us to follow the intrinsic apoptosis pathway in Arachidib-1 treated cells. These findings show that Arachidib-1 should be further investigated as a potential treatment for triple-negative breast cancer. Thank you for listening.